on phase separation. Please, Julie.
So thank you very much to the organizers for the opportunity to present today. So I wanted to initially acknowledge all the people in my group whose work I'm going to speak about, including Brian Sang, Tae-Hoon Kim, Alaji Ba, who's now a professor in Syracuse, Robert Vernon, Eva Prudenashak, and Yishuan Lin, my collaborators at SickKids, Luyang Wang and Steve Scher, at McGill University, Nachum Sonnenberg, and at the University of Toronto, Louis Kay, Husan Chan, Alan Moses, and Ben Blanco. So today I want to tell you about our studies of what I like to think of as a model neuronal granule composed of the low complexity regions of FMRP and Chaperone in order to study translational regulation. And these two RNA binding proteins have C-terminal low complexity IDRs that have RGG motifs that enable dynamic protein and RNA interactions, and they both readily phase separate with RNA. We chose them because they're very highly expressed in the brain. They bind RNA, are involved in mRNA transport and stability. They're known to be translational regulators. And importantly, their mutations are linked to autism spectrum disorders, and knockout or loss of function decreases synaptic plasticity in neurons and recapitulates neurodevelopmental behaviors in mouse models. They co-localize in cells, in particular in P-bodies and stress granules, and of interest to us in neuronal mRNA transport granules, which take RNA from the cell body in a translationally silent state towards the synapse, where upon activation, the granule transiently disassembles the RNA that becomes available for translation, and the proteins that are made are learning and memory, often cytoskeletal proteins that remodel neuron-neuron contacts, as well as neuronal receptors. And then immediately, these granules reform. And so we hypothesized that synaptic control of the critical concentration for phase separation enables this reversible assembly and disassembly of neuronal transport granules to regulate this activity-dependent translation, and that in disease, the granule composition and properties and the assembly-disassembly dynamics are perturbed. So we are using in our model system just the C-terminal low complexity regions because they are necessary, as you can see. If you delete them, you no longer get phase separation, but they're also sufficient. So just having the isolated C-terminal region gives rise to phase separation. They also mediate RNA binding at 100 nanomolar or hundreds of nanomolar KD, and in general, fairly non-specifically, although for most of our studies, we're going to be using SC1 RNA, a synthetic G quadruplex forming RNA. So what about synaptic stimulation could change the properties of phase separation? And it's known in the literature that multi-site serine threonine phosphorylation of FMRP by casein kinase 2 represses translation, and that stimulation of the neuronal receptor mGlu-R leads to dephosphorylation of FMRP by PP2A, and that initiates translation. And that this activity-dependent translation is linked to transient FMRP dephosphorylation is followed then by rapid rephosphorylation. So in vitro, we can take casein kinase 2 and phosphorylate the FMRP low complexity region, and we get multi-site phosphorylation that enhances the phase separation propensity. With this phase diagram, you can see needing less RNA and less FMRP to phase separate than the non-phosphorylated case. And this is consistent with theory that predicts that charge blockingness promotes phase separation, with phosphorylation increasing density of negative charge clusters with this sliding average charge plot. On the other hand, arginine asymmetric dimethylation by PRMT1 disrupts phase separation, as does arginine to lysine mutations, although it doesn't change RNA binding. And we can we can use this enzyme and get 6 to 8 arginines modified. So here's the test of the hypothesis. Using a rabbit reticulocyte lysate in vitro translation assay with luciferase mRNA so that we can read out luminescence as our measure of translation, and plotting here the inhibition of translation, adding the FMRP LCR, you can see nothing, 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 and then boom, we have a switch with a Hill coefficient near 5, as opposed to adding puromycin with a Hill coefficient closer to 1. And we need less phosphorylated protein to affect this switch to translational inhibition, and more arginine methylated protein. And if you take some of this assay and look in the microscope, you can see that all of the conditions that lead to translational inhibition have droplets under the microscope, and all those that don't are fairly clear. So this temporal match of FMRP phosphorylation states with disassembly and reassembly of neuronal RNA granules, and the correlation between translational inhibition and FMRP phase separation, as well as predictions of phase separation and known phase separation of other proteins linked to synaptic plasticity, support this model that phase separation of RNA binding proteins underlie assembly and disassembly of neuronal RNA granules and activity-dependent translational regulation. But regulation of translation also requires control of RNA stability, and translation and deadenylation are tightly coupled. So we're going to add Chaperone now to our model system because Chaperone is associated with deadenylation. And FMRP and Chaperone LCRs phase separate in a manner that's dependent on FMRP serine threonine phosphorylation. And we're going to use NMR to look at the molecular interactions within condensed states of FMRP and Chaperone. So here we have some spectra of labeled Chaperone 1 LCR in buffer in black, and partitioned into the condensed phase of FMRP in green, or partitioned into a condensed phase of phospho-FMRP in pink. And you can see that there is broadening of the resonances in the condensed phase, as well as some small chemical shift changes. And you can see more significant broadening in the pink PFMRP with the loss of the resonances. And you can see that arginine, tyrosine, and phenylalanine peaks in particular show significant broadening and chemical shifts. And we can quantify the broadening of the Chaperone resonances by looking at the intensities in these condensed phases divided by the intensities in the buffer. And we look at this intensity ratio. And you can see that there's significant loss of intensity in the condensed phases away from the edges. And this is reproduced, this intensity ratio plot is reproduced again, and I've just colored these regions, arginine-rich regions, and you can see that those are more different in between the FMRP and phospho-FMRP, and here's just a difference, than the aromatic regions in the pale pink here, which are much more similar. So the differences in the arginine-rich regions are likely sites of differential charge interactions of the arginines with the phosphates, whereas in the aromatic rich regions, this reflects common interactions to both. These tyrosines that are in the aromatic regions are known to be phosphorylated, and we can in vitro phosphorylate them using the F4A kinase. And with multi-site phosphorylation, we can see, similarly to serine threonine phosphorylation of FMRP, increasing propensity to phase separate with unphosphorylated Chaperone, the tyrosine phosphorylated Chaperone increases the propensity to phase separate with non-phosphorylated FMRP. But when both are phosphorylated, you're lowering that propensity to phase separate, likely due to charge repulsion. So when we now add RNA to this phosphoserine threonine FMRP and non-phosphorylated caprin, what we see is the appearance of two phases. So we have a multi-phase condensate, subcompartmentalized droplets. That doesn't happen in the tyrosine phosphorylated caprin, non-phosphorylated FMRP case, where we still see a single droplet. So this complex phosphorylation switch, depending on FMRP phosphoserine phosphothreonine modification and caprin-1 phosphotyrosine phosphorylation, is suggestive of how integration of different signaling pathways can control translation and RNA processing in cytoplasmic RNA bodies. And this multi-phase situation is dynamic, and we can observe as a function of time that the outer FMRP, phospho-FMRP phase fuses and the inner caprin-1 phase fuse. And these are similar to dynamic coexisting multi-phase compartmentalization of cytoplasmic stress granules and nucleoli with these high resolution images. With lower resolution images of the neuronal granules, we're not so sure, but if you squint, you can sort of see that the caprin-1 and FMRP are not totally co-localized, with the caprin-1 perhaps being in the inner phase and the FMRP in the outer phase, similar to our model neuronal granule. So what about deadenylation biochemistry? So we can use the C07 catalytic unit of the CCR4 NOT complex and use a poly-A RNA with 38 A's and use polyacrylamide gel electrophoresis to monitor the progress of the reaction. And in these multi-phase droplets, with phospho-FMRP and non-phosphorylated caprin, we see more rapid deadenylation kinetics than in buffer, whereas with the opposite phosphorylation pattern, we see similar kinetics to buffer. And in these multi-phase droplets, you can see with labeled RNA as a function of time that inner compartment disappearing, consistent with deadenylation occurring within that compartment. We can also look at the deadenylation rate in caprin condensates without added FMRP. And using ITC, we can see that initially adding caprin to C07 gives rise to an ITC signal, and then a second signal related to phase separation. And if we look at the rates of deadenylation at those molar ratios, so this is basically buffer, this is at the 1-to-1 binding, and this is in the phase separated caprin condensate, you can see that binding of caprin to C07 doesn't enhance the kinetics, but being in a caprin condensate does. And we're currently testing a hypothesis that the positively charged protein solvent environment is what leads to the phase separation dependent rate enhancement by affecting C07 conformational equilibria. So how does phase separation affect the biochemistry of translation? This is a little easier to consider with just differential partitioning of the translation machinery due to phase separation, with RNA being separated from other components. And in this case of our multi-phasic droplets, you can see that the RNA is in the inner caprin phase, and EIF4E cap binding protein is in the outer phospho-FMRP phase. So we can do translation and deadenylation assays as a function of different FMRP and caprin composition and phosphorylation states, and you can see over 4 orders of magnitude regulation of the translation rate with this log scale, and just a single factor of 10, a single order of magnitude control of the deadenylation rate. With the rates being inversely correlated, as you would expect. And so phosphorylation appears to act as a switch to control the phase behavior and the subcompartmentalization, as well as the enzymatic activity. So with this in mind, we joined a large collaborative team led by Ben Blanco to look at an autism-associated neuronal microexon that has activity-dependent splicing in the translation initiation factor EIF4G1's IDR. And this microexon of 8 amino acids promotes interactions with multiple neuronal granule components, including FMRP, that lead to a reduction in translation as observed by polysomal RNA measurements. But we can see in vitro that the addition of the microexon leads to enhanced phase separation with RNA as well as with phospho-FMRP compared to without the microexon. And so disruption of this microexon splicing, which is often detected in autism, is expected to contribute to cognitive impairment by dysregulating the neuronal activity-dependent translation outputs, and this has been supported by electrophysiology and mouse behavioral studies in this highly collaborative paper. So what can we generally infer regarding the impact of IDR phase separation on biology and disease? This is an important question considering that 60% of proteins in the human proteome have significant IDRs, and about 20% of the proteome has IDRs making up about half or more of the protein. And so doing some bioinformatics using P-score, which is a predictor of phase separation that our lab developed, that predicts IDR-driven phase separation based on long-range planar pi-pi contacts using PDB-derived sequence statistics. We can see that in the human proteome, we have over 4% of human proteins predicted to phase separate via this mechanism, but autism-associated proteins and cancer-associated proteins are predicted to be much more likely to phase separate. And this is not comprehensive. Just looking at pi-pi-driven phase separation, we are developing a new predictive algorithm that takes into account all biophysical interactions, and that manuscript is now in preparation. But it's not surprising that autism and cancer-associated proteins have this higher propensity to phase separate, since we are increasingly being made aware that biomolecular condensates regulate many processes that are implicated in autism and cancer, from chromatin condensation all the way to signaling. And in the case of autism, we can specifically talk about activity-dependent transcription and splicing, packaging into neuronal transport condensates, transport towards the synapse, then activity-dependent translation, which I talked about today, synapse remodeling, receptor clustering at the synapse, and then signaling condensate transport back to the nucleus. In the case of cancer, you can substitute this synapse organization for overactivation of growth factor receptors, and you can see very similar processes involved. So I think there's going to be a lot to learn about the regulation of biology through condensates, and I'm just going to stop there and acknowledge the members of my lab and collaborators, in particular Thomas Ganatopoulos-Pernatsis, Ben Blanco's postdoc, who led that collaborative work, and thank you.
Thank you, Julie, for really exciting presentation. I'm sorry that we don't have time for questions, and we will hear more today and tomorrow about the connection of human disorders and phase separation. Thank you so much. So we are moving to the next speaker, Reut Shalgi from the Technion, and she will tell us about mutant FUS aggregation protection as it's revealed by Chaperone network screens. Please, Reut.
Thank you, Nassim. Let me share my a screen with you. Okay, so first of all, I'm really happy to be here, and this is an excellent lineup of speakers. Today, in my talk, I want to take you to the dark side of molecular condensates and phase separation, and talk about a really exciting project that we have here in the lab, revolving around a mutant FUS aggregation protection by chaperones. So pathological aggregates are really a hallmark of a myriad of neurodegenerative diseases. Some of them are shown here. They include Alzheimer's disease, Parkinson's disease, Huntington's, as well as ALS. And what is we know today about the pathological aggregates of the vast majority of cases in ALS is that they're actually forming through the process of phase separation and aberrant phase separation. So I want to talk to you about stress granules, and stress granules are actually an example for a dynamic phase transition that's actually part of normal cellular physiology. It's a liquid phase separated membraneless organelle that's generated dynamically in response to a variety of stress conditions. And then it dissolves back in the recovery stage when the stress is stopped. It's known to contain multiple RNA binding proteins, some of which are listed here, for example, TF, FUS, and TDP43 and others. And its actual physiological role is thought to be to protect RNAs from a translation during stress conditions and then release it back to the translating pool in the recovery stage. So what happens in a disease, and in particular in ALS, is that aberrant phase transitions are occurring for parts of these proteins that form the stress granules, and they lead to this more solid-like compartment generation that's actually accelerated by disease-related mutations. And what is special about this compartment, this solid phase transition, is that it cannot be dissolved back into its original phase. And it's actually accumulating as a pathological aggregate, and it's thought to be a major contributor to the ALS phenotype. Now, the way that a major thing that we need to consider in this process, both during the generation of the liquid-like phase-separated organelle, as well as in the solid-like, is that it involves conformational changes from the diffuse state to the liquid-like state, and from the liquid-like state to the solid state, what we often call a protein misfolding. And when we talk about protein misfolding, we immediately think of the master regulators of protein folding in the cell, the molecular chaperones. So just to remind everyone, molecular chaperones in humans comprise a network of over 300 members that are separated into different families. And they're known to recognize misfolded proteins and act to unfold them and refold them in a catalytic cycle, or promote their degradation upon terminal misfolding. One of the major chaperone families that's very important and forms actually a subnetwork within the chaperone network is the HSP70 family. Now, HSP70 is a highly abundant and fairly promiscuous chaperone that is known to recognize misfolded proteins and act to refold them. And it requires the assistance of two co-chaperones, an HSP40 or a DNAJ, and a NEF. And while this catalytic cycle is highly conserved from bacteria to human, actually these families are undergoing a major expansion in evolution. While the HSP70 family in humans comprise of 13 different members, the NEFs have 14 different members in humans, and the DNAJ family has actually undergone the most massive expansion with over 50 different members in humans. And this really raises a very nice concept that we'd like to think about of functional diversification between within the different chaperone families and how they contribute to regulation. And this is an issue we know very little about. Now, when we look at these aberrant phase separation transitions in ALS that are occurring from the liquid-like to the solid-like pathological aggregates, what we would like to know is whether we can identify any chaperones that can intervene within this process and prevent this aberrant phase transition. And this really brings me to the question of today's talk that we decided to ask. First of all, can we generate a framework that will allow us to systematically examine the role of each chaperone in the network in the modulation of pathological aggregation, and whether we can do that for classical aggregates, as well as the phase separating aberrant aggregates that are observed in ALS. And the short answer is yes, we can. And I want to tell you the long story now. So we decided to start with a fairly well-characterized pathological aggregate that's an amyloid type of aggregate, the polyglutamine huntingtin that's mutated in Huntington's disease. And here we're expressing this huntingtin with a long polyglutamine stretch tagged to GFP in a cell line, HEC293 T cells. And upon 48 hours of expression only, we can see that a fair amount of the cells are already expressing these bright foci that correspond to protein aggregates. Now, not all of the cells are expressing these aggregates, and we're going to take advantage of that fact. In the meanwhile, the wild type form with a short polyglutamine is forming this diffused fluorescent pattern in the cells. So we decided to actually quantify that, and we're using a highly quantitative and reproducible FACS-based assay called PULSA, which basically allows us to determine using the width and height of the fluorescent peak and discern cells that have a diffuse fluorescent pattern from cells that contain an aggregate. And this is what it looks like, the raw data. You can see here that the wild type form of Huntingtin forms a single cell population of fluorescent peaks, while the mutated Huntingtin forms an additional cell population over here with different fluorescence characteristics that actually correspond to cells containing aggregates. Now we want to turn this to a readout for a screen. And here I'm showing you our control for the screen, the co-expression of Huntingtin polyglutamine with an unrelated protein, DS-RED, and our basal rate of aggregation is about 34%. When we co-express instead a chaperone DNA JB8, we get a marked reduction in aggregation to about 13%. And on the other hand, when we co-express HSP90, we get actually an elevation to about 43%. So we can actually make this measurements into a score that we term aggregation modulation score, which is simply the log ratio between the percent of aggregate containing cells in the presence of the chaperone normalized to the control. And as you can see here, DNA JB8 got a negative score and HSP90 got a positive score, and they're both highly significant because they're crossing this 95% confidence interval. Now, this is the result of our screen of 66 different chaperones. They're color-coded according to their different family they belong to. And what you can see here that we got a continuous spectrum of aggregation modulation scores. Interestingly, on this side, you can see that over a third of the chaperones actually confer aggravation of the Huntingtin polyglutamine aggregation phenotype. And on this side, we've identified 4 chaperones that provide significant protection from aggregation of Huntingtin. Now, several of these have been shown before to affect Huntingtin aggregation, including HSP90 and DNAJ-A1 on this side. And in the protection side, 3 out of the 4 that we've identified have been shown before to affect aggregation of Huntingtin, to protect aggregation. And this was made us actually very happy because it means that this framework is actually working and doing what we want to do. We were able to identify another 4th chaperone that's significantly protected from Huntingtin aggregation. This is DNAJB12 short. And here's a schematic representation of the domain architecture of DNAJB12. And as you can see here, it's actually an alternative isoform of the canonical full-length DNA JB12 isoform shown here. And it lacks the J domain that's known to be responsible for HSP70 interactions. And with co-IP, we actually verified that indeed the full-length isoform has strong interactions with HSP70 while the short one barely interacts. And as you can see here from the aggregation modulation scores, the short isoforms provide significant protection from Huntington aggregation, while the long one, the full-length one, aggravates the phenotype. So now we know that we have a working framework. We want to shift gears to look at the phase separating aggregates in ALS. And we chose to look at FUS, which is relatively unexplored in this context. So a myriad of mutations have been identified within FUS to cause ALS. And we decided to concentrate on a bunch of them here at the C terminus of the protein that reside within the NLS region. And these lead to a cytoplasmic perinuclear aggregates, as shown here by confocal microscopy, as opposed to the wild-type FUS that generates this diffuse pattern in the nucleus. Here is the result of the screen of the 66 chaperones for FUS modulation, and you can immediately see that it's very, very different than the result that we got for Huntington. And I'll point you to a few key differences. First of all, in the aggravation side, we only have 2 chaperones, and the rescue side, we have many more, and the magnitude of a rescue is much larger. We verified some of them using the fluorescence microscopy, and we recapitulated the results. And when we compare directly between the aggregation modulation score in Huntington versus mutant FUS, you see that there's basically no correlation, and the identity of the rescue chaperones is completely different. And this is actually very exciting because it means that the 2 pathological aggregates are very different in their requirements. And now I want to focus on one of them that we characterized in depth. This is DNA JB14. This is a structure of DNA JB14. And surprisingly, or maybe not so much, similarly to B12 that I've shown you before, this one also has a naturally occurring short isoform shown here, and it also lacks the J domain. And as you can see here by co-IP, indeed this short isoform does not bind HSP70 as opposed to the full-length one. Using immunofluorescence imaging, we saw that in the presence of it was called HPD mutant, and as you can see here by co-IP, it indeed did not interact. Using IF, we saw that this mutant and an additional mutant had a fair amount of FUS aggregation in them. And when we quantified this by FACS, we saw that indeed they both lost the ability to protect from FUS aggregation. This led us to the question of what's the role of the complex in aggregation protection? Or more precisely, is it the complex that's doing the job, or maybe the 2 chaperones independently? So in order to answer this question, we performed the following experiment. We introduced B14 full-length into cells, either with an SI control or together with an siRNA to the endogenous B12 that we were able to knock down to about 50% of its level. And what you see here is that upon this knockdown, B14 full-length loses its ability to protect from FUS aggregation by about 50%. So we conclude from that it is really indeed the complex that's responsible for the protection phenotype. So these and other experiments have led us to propose the model whereby B12 and B14 interact both together and with HSP70 to perform aggregation protection of mutant FUS in ALS, whereas the short isoform of B14 is unable to form the complex nor to interact with HSP70, and it does not protect from FUS aggregation. And finally, we wanted to see whether we can recapitulate this aggregation protection in primary neurons. So to that end, we worked with neuronal cultures that we infected with either wild type or mutant FUS, as shown here. So in neurons, actually, mutant FUS generated this complex phenotype of aggregation, whereby, as opposed to 293 T cells, we don't see only one large aggregate, but rather several large aggregates with a smaller foci within the cytoplasm, and they were devoid from the nucleus. So we decided instead of FACS to use image analysis, and we had a classifier that was trained to distinguish between this phenotype and the wild type phenotype that was nuclear. And the next step was to co-express either the full-length isoform or the short isoform of B14 in these primary neurons together with mutant FUS. And our image analysis indeed showed that full-length isoform of B14 was able to provide substantial protection for mutant FUS aggregation in primary neurons compared to the short isoform. So here I'd just like to summarize that we've come up with a framework to test the functional effects of virtually any factor on any pathological aggregate, including the phase separation aggregates, such as FUS. This led us to identify a complex composed of DNA-J, B14, B12, and HSP70 that provided significant protection for mutant FUS aggregation. And on top of that, we've revealed that naturally occurring isoforms of J proteins have actually different functional effects with respect to different types of aggregates. And we're really excited about this because this really means an increased level of complexity to the chaperone network conferred by isoforms and their interaction between them that has differential effects on different pathological aggregates. And in the end, I would like to thank the lab and especially the two exquisite grad students who did this work, Kinneret and Amal. They were helped by Lior with image analysis and our collaborator in Shai Berlin's lab who helped us grow primary neurons. And I'd like to thank you for your attention.
Thank you so much for a very interesting presentation. I apologize to move ahead. And the next speaker is Eran Hornstein from the Weizmann Institute. And the topic of his presentation is spatio-temporal proteomic analysis of stress granule disassembly. Please, Eran.
Thank you, Nissim. Also, Eran and Roger for the invitation. It's a great pleasure to be here. So I'll talk today about a project that was done in collaboration with Paul Anderson, Nancy Kadersha, and Pasha Ivanov in Harvard Medical School. Tammy Geiger from Tel Aviv University was the driving force behind the mass spectrometry analysis we are doing. And then Ludo Vandenbosch that's assisted with fly analysis together with Tom Mons. And in front of you, you can see a graphical summary of the work. So basically we focus on stress granules that has been mentioned before. These are cytoplasmic condensates of RNA binding proteins and RNAs that are arrested from translation. And quite a lot is known about stress granule assembly, but not much is known about their disassembly process. And some of it has to do with limitation in characterizing the composition of stress granules, particularly during the stepwise process of disassembly. And this is exactly what we tried to do. So you can see a stress granule inside quite a few proteins in multicolors. One important point is that we believe, based on data that I'm going to share with you, that additional proteins depicted here in red, these are disassembly engaged proteins, come in during the disassembly, but not before, and they are essential for allowing disassembly to go on. Furthermore, we were able to compare the disassembly process under normal cellular conditions in simple human U2S cells to what happens in a cell culture model that mimics, at least in part, a neurodegenerative disease that Ruth just touched upon called amyotrophic lateral sclerosis, or ALS. So I'll start with this, and I'll mention Chagai Marmor-Colet, who is a PhD student in my lab and the driving force behind the work and is an outstanding scientist. Okay, so we have been using a technology developed by Alice Sting to profile the proteins in stress granules, in membrane-less organelles. And the technology is based on introducing a bait that is normally localized into a given compartment with a peroxidase activity that's allow radicalization of biotin phenol and tagging by a biotin moiety, the biomolecules at the proximity of the bait. And altogether, it allows to stain defined compartments with biotin. So that's a graphical image that would help you to bear in mind how the biotin staining is done in live cells, and later after lysis, all the biomolecules in a given compartment are already conjugated to biotin and are subjected to downstream mass spectrometry analysis. The apex proximity labeling approach have been used for very extensively, initially in compartment restricted domains where there is a membrane surrounding it, but progressively into smaller and smaller and more dynamic compartments, sometimes that are those that are non-diffusion restricted, such as synaptic clefts, G-coupled protein receptors, and lately also to membraneless organelles or RNA granules, including in the work of Sebastian Markmiller with Jin Yao, who is here because they were the first to use this technology for stress granules, and also in the work of Padron et al. All very interesting works that provide substantial evidence for the ability to profile tiny diffusive compartments, given that you do it carefully enough and with sufficient controls. And here is the first slide from our data. You can see here the apex peroxidase driven to stress granules in cells by FXR1 and the nice colocalization with the TR1 marker. But this works only with stress, because without stress we do not see any microscopic signal. FXR1 is one bait that is directed with very high partitioning fraction into stress granules. About 80% of the FXR1 is in stress granules on stress conditions. FMR1 or FMRP is another such protein, a partner and a binder of FXR1 that Julie Forman-Kay just mentioned a few talks before me. And then G3BP1 is perhaps the most established marker of stress granules. And all have very nice partitioning fraction, and you can see how they tightly colocalize with stress granules. To do the analysis properly, we use those three baits, multi-bait apex approach, and analyze the data with cross-reference between the list we gained from those three baits. In addition, we were having also the cytoplasm as a reference that we always subtracted. So data that we have is always with reference to the surrounding cytoplasm, the solvent around the phase-separated stress granules. Here is a volcano plot that gives you a glimpse on the data. So to the right is anything enriched in stress granules, and to the left, enriched in the cytoplasm and relatively depleted in stress granules. Blue features are a benchmark list of proteins that are known to be excluded from stress granules, and they are elsewhere in the cytoplasm. The red features are known stress granules marker, and these are enriched in our data, and that is, of course, very reassuring. The black features are our discovery capacity. So these are new stress granule proteins that we have discovered, and we were able to cross-reference these with our three baits with a stepwise confidence and decreased numbers. I'll focus primarily on what you see in the middle of the Venn diagram, proteins that were depicted by two markers, roughly 240 proteins, 45% of which are novel and were not discovered before in stress granules. Our list talks nicely to previous omics analysis of proteins in stress granules, including analysis by Jin Yao with a single beta apex, a fractionation analysis of Roy Parker, the pre-bio-ID approach. And again, you can see how we have things that are known, and that's reassuring, and new discovery capacity. The proteins that we identified are typical of what you could expect from stress granule proteins. They have high percentage of intrinsically disordered domains. So you can see here in green, these are the proteins we have identified, and yellow, blue, and red are the same color codes for the proteins that were described in the literature. And a similar analysis was done also with P-score that Julie K. Forman has reported. Using unsupervised clustering of the proteome, and a heat map you can see here tells us that the proteome of FMR and FXR are almost identical and cannot be distinguished. They are naturally or expectedly distinguished from the cytoplasm at the upper left corner, but quite surprisingly, the G3BP1 proteome is distinguished from both, as if it generates a compartment of its own. And the idea of two compartments was again mentioned before by Julie, and I would like to show you similar data also by principal component analysis. So the proteome shifts a tiny bit for the cytoplasm upon stress. It shifts to more dramatically for the G3BP1 proximity proteome. The FXR and FMR proteomes are quite similar, and both are distinguished from the G3BP1 one. All right, so the idea that we have two compartments needed substantiation by an orthogonal approach, and to this end we used super resolution microscopy. What you can see here is storm microscopy of FXR1 and G3BP1, suggesting that they are in two compartments, and those compartments are further set apart as two substructures, whereby the G3BP1 is the core of the stress granule, here more quantified in the center, and the FXR is at the periphery as a shell. And this is storm microscopy. STED microscopy gave a similar pattern. The focus is not great, but you can imagine how the core here is surrounded by a shell of FXR. Furthermore, those two have different biophysical properties, which is again interesting, because you can imagine that the proteome defines the diffusion and additional biophysical properties. G3BP1 compartment is highly diffusive and recovers after photobleaching really quickly. But FXR1 is way less diffusive and did not recover even hundreds of seconds, even a few minutes after the photobleaching, suggesting different properties for the same organelle, same stress granule with two tiny compartments. Going next, we wanted to understand what happens in the disassembly. And again, the Apex technology allowed us to understand things that were not known before. What is the composition and how it changes in the disassembly? So you have stress and stress granules, and now you wash the stressor, and in recovery, those stress granules disassemble progressively. If we look now at the corresponding proteome, you see that there are many proteins that are known or newly discovered stress granule proteins, and they progressively dissolve. So you get less and less of them until at roughly 100 minutes, you don't have too many proteins at the proximity of the bait. Furthermore, we identified another very interesting element. Other proteins get engaged only upon the cue to disassemble. So in correlation with the washout of the stressor, new proteins come in. And we thought that these are disassembly engaged proteins, and some of them might be functional and hence interesting. We checked only a handful by a functional experiment using knockdown and checking the implication of knockdown of a few of those proteins to the disassembly. And you can see that indeed some of them affect the disassembly process. Two were particularly interesting, and we followed on them up further. These are two conjugating enzymes in the SUMO pathway, a pathway that is reminiscent of the ubiquitin pathway, only smaller, and conjugating a peptide that is called SUMO. So take a look here at a diagram of the SUMO pathway. It has an E1, E2, and sometimes also uses E3 conjugating enzymes. It's so simple that it has only one E2 enzyme, and its inhibition is sufficient to block the pathway completely. The SUMO pathway component seems to be engaged with stress granules more than in the cytoplasm, which is interesting and rather novel. And we could support this also by Western blot analysis. Some of those components, like the E2, UBE2I, also called UBC9, are seen primarily upon disassembly. and the inhibition of the pathway by small molecule inhibitor of simulation blocks stress granule disassembly in a concentration-dependent manner, suggesting that this pathway is functionally relevant for the process of disassembly. The proteins inside stress granules are enriched with reported simulation sites by analyzing or correlating the proteins we have identified to those reported to be simulated. And when we asked for at least one of them, how simulation affects the regulation of stress granule disassembly in cis, we took the FMRP and point mutated 2 known simulation sites, so it was simulation dead, and indeed it affected or inhibited the process of disassembly. We do not intend to imply that FMRP is the most important simulated protein or the only one, of course, but it tells us that even just one particular protein is sufficient to drive an effect in cis downstream of the simulation pathway. And altogether, I'll take you back to part of the diagram. We think that sumo ligases are disassembly engaged proteins, and they are essential for the process. When our work that is now soon to be out in a molecular cell was in revisions, the group of Stefan Muller in Heidelberg published a very interesting and related work talking quite similarly about how sumo pathway regulates stress granule disassembly. And those together, I think, support one another in the idea that sumo is critical for disassembly. I'll take you in the last few minutes to see pathology. So again, we are interested in ALS, took a model different from what Reut has talked about, and this is the C9orf72 D-peptide repeats, a simple cellular model that recapitulates parts or facets of the molecular phenotypes seen in the most prevailing form of ALS. Those D-peptides are seen in the nucleus of cells when we use a particular version called proline-arginine repeats. They are part of a bigger family of 5 different versions of those D-peptides. They are toxic to neurons and they drive ALS in humans. When we put those D-peptides in cells in culture, what you can see that the disassembly process does not proceed normally. In control, the stress granules disassemble after a while, but in the PR50, proline-arginine 50 repeats, we see the stress granules keeping on. And that was reported also by others before by microscopy, but we thought that it is a particular opportunity for us to use the apex proximity labeling. So quite a long time after disassembly is accomplished in control conditions, we still see those stress granules. And we ask, what's the difference in the composition? So we compared the ALS model to control model, and again did mass spectrometry after apex proximity labeling. You can see that many of the disassembly engaged proteins do not engage, suggesting a potential mechanism to why the PR50, the ALS model, might inhibit disassembly. We also see proteins that should not be there but are residual in what we believe is an aggregate that remains after disassembly is completely accomplished in the control conditions. But this is not thoroughly studied yet in our group and is quite interesting. The sumo conjugating enzymes among the disassembly engaged proteins are also not properly engaged and do not convey appropriate simulation, which could be seen in the ALS model as decreased simulation of the stress granule proteome. So for this Western blot analysis, we pulled down by apex the proteome of stress granule and subjected it to Western blot analysis and depiction of sumo. And there is always a drop in the simulation level with the PR50. Finally, with Ludo Vandenbusch and Tom Wan, we asked whether in vivo, in a fly model, we can see genetic interaction of the ALS model with simulation, whether it can rescue it. So you can see a necrotic eye of Drosophila with those nasty necrotic stains in the eye, and overexpression of the UBE2 equivalent of the fly LWR recovers largely the eye phenotype, and this is quantified here. So the idea that's in vivo in a simple neurodegeneration model in the fly, the overexpression of SUMO can recover an ALS-like phenotypes, talk to the relevance of simulation also in ALS models, which we of course need to further explore using mammalian models and human neuropathology. I'll give you a quick summary. We use multi-bait apex. It's a method that allows cross-validation, increasing the signal and decreasing the noise, especially when dealing with membrane-less, highly diffusive organelles. Novel stress granule proteins and distinct substructures were discovered. Disassembly engaged proteins seems to be highly relevant to regulating facets of the disassembly process. Stress granule composition and disassembly engaged proteins are impaired in ALS conditions, and SUMO controls stress granule disassembly and is dysregulated in ALS. I'll skip the graphical summary again. Thank the group. Chagai, Aviad Siani, who is an equal contributor on the work, and many others from the group, Tammy Geiger from mass spectrometry, Paul Anderson, Pasha Ivanov, Nancy Kedersha, Ludo Vandenbush, and Tom Mons. And I thank you for listening, and we'll be happy to take questions if there are any.
Nissim, you're muted.
Thank you, yes. Thank you, Eran, for really fantastic talk. And because of our tight schedule, we'll not have time for questions, and we will move on. And we are really very fortunate to have Stephen McKnight from the UT Southwestern as the last speaker of this exciting session. And Stephen will tell us about human genetics of neurological disease as a guide to the structure and function of low complexity domains. Thank you, Stephen, for joining us.
Well, thank you, and to the organizers of the meeting. This is my first Zoom presentation ever, so I'm hoping I can pull it off. Can people hear me? Just give me a nod if it's working.
Yes, it's working.
We can hear you fine. Thank you.
Okay. I thought I'd start this talk off with a discussion of these interesting cancer-driving fusion proteins that take place when a reciprocal translocation event covalently links a specific region of the FUS or EWS proteins, and that's this squiggly line on the left, to the DNA binding domains of a number of different transcription factors. So these translocations create fusion proteins that, of course, are composed of two components, a DNA binding domain on the right and the squiggly low complexity domain on the left. And these are the driving events for many terrible forms of cancer. So what we've done over the years is to study gene expression, study transcription, and try and understand, for example, how these proteins work. So on the right-hand side, with respect to leucine zippers or zinc fingers or ETS domains or homobox domains, 20 to 30 years ago, it was possible to understand how these domains work, how they facilitate the attachment of a transcription factor to a specific region of DNA. So this is largely solved, the right-hand side. The left-hand side, though, has been a mystery. There's no question that transcription factors rely on these squiggly domains, these disordered domains, in order to regulate transcription, to either activate or repress. They were originally discovered in the context of transcriptional activation, but how they work mechanistically has always been a mystery. So I followed this and have not pursued it. I kind of took 20 years off, but completely by accident, about a decade ago, we made an observation. Let's see. My slides are not advancing for some reason. Do you know why my slides aren't advancing?
I was gonna say, click the start video so they can see you.
They can see me, but my slides are not advancing. I'm sorry about this, you guys. Are they advancing now?
Try to stop your sharing and reshare it again. And when you share, don't share the screen, but the presentation itself. It gives you an option to share only the presentation, slide presentation, rather than the screen itself.
Now, how do I advance?
Yeah, great.
How do I advance?
Just click on the slide.
Click on the slide. Okay, you guys, again, please accept my apologies. So with the help of some of my trainees, I may be able to advance the slides now. But anyway, about a decade ago, we stumbled over to this kind of crazy observation that the purified low complexity domain of the fused and sarcoma protein, fused protein, would phase separate.
There's a video so they can see you. Show them.
Sure. This is I knew this would happen. I'm really sorry, you guys. Come on. Look, they don't need to see me. I don't know what's wrong. But let me just proceed without you being able to see me. It doesn't really matter. All right, let's get back to this. We stumbled over this crazy observation that the purified low complexity sequence of fused would phase separate out of aqueous solution to form either liquid-like droplets or these more solid hydrogels. And you've heard lots about this, and this has become a very, very exciting field. The topic of this meeting is all about phase separations, and I think it's very important to biology. We're going to learn lots and lots from what's going on and what you're hearing about. But my own interest is very limited, very myopic. And that is, I wanted to know what's happening. What are these protein sequences doing precisely in this process of phase transition? Because I expected, at least I hoped for the possibility that it would tell us something about the function of these gibberish looking sequences. And as I said earlier, the function of these sequences has historically been a mystery. So now what we've got is we've got these gels or we've got these droplets, and they create an assay. And the initial assay that was created by Masato Kato in the lab about 10 years ago was to make these little gel-like droplets, put them in a depression slide so that they could be viewed by confocal microscopy, and then test them. Do binding assays. So in the top part of this slide, what you can see is the hydrogel droplet has been challenged with soluble GFP and there's no binding. However, if the GFP is fused to the same low complexity domain that created the droplet, now you see avid retention of the fusion protein by the droplet. So this has become our assay, a very simple binding assay. There's specificity to it, and I'll go into that to some degree, but what we imagined is that there's the possibility is this binding assay might tell us something about how these particular protein domains work. That was the hope. So what we need to know is what's in this droplet. What's the droplet composed of? So by electron microscopy, we quickly observed that the droplet is chock full of homogeneous polymers. That's shown on the left of this slide. They're very much amyloid-like polymers. And if we lyophilize the sample and shoot it in the X-ray beam, we see diffraction rings at 4.6 and 10 angstroms. And so between the electron microscopy and the X-ray diffraction, we can rapidly know that what we've got here is an amyloid-like polymer. But interestingly, the polymer is unlike traditional amyloids that are irreversible. So the SUP35 protein that Reed Wickner has studied in the context of yeast prions is shown on the left of this agarose gel, where we're exposing the SUP35 protein to different amounts of sodium dodecyl sulfate. And what you can see is that the polymers made from the SUP35 protein are extremely stable. Even in 2% SDS, they won't come apart. On the right-hand side is an image of a gel in which we've, instead of running the prion, the yeast prion, we've run polymers made of the HNRMPA2 low complexity domain. And what we notice in that case is that the polymers disassemble as soon as they're diluted. So there's some fundamental difference between the polymers on the left that are irreversibly assembled and are highly stable, won't come apart, and the polymers on the right that are very labile. Now, I'm not going to go into the details of this because Rob Tico is talking later in this symposium, and Rob and I and others have studied the structures of these polymers, and we think we have an idea as to why the polymers formed from low complexity domains are labile, but I'll leave that to Rob. So what we needed, we've got an idea, and the idea is that the interactions that are creating these gels are specific and may be of biological importance, may not be. So what we needed was a biological system with two correlates. One correlate would be that the low complexity domain involved in that biological system would be functionally required. You could show that it was needed to achieve something. The second correlate was we needed to be able to study the structure of that low complexity domain in the proper biological assembly. So if I could do this with RNA granules, I would have done it a long time ago, but I can't reconstitute RNA granules to study the low complexity domain of FUS or HNRMP in the granule because I can't reconstitute it. But luckily, with intermediate filaments, we're able to do this. So what I show on this slide is a schematic diagram of intermediate filaments. These would kind of be the last proteins I would have ever thought I would be studying late in my career, but they fit the correlates. They gave us what we were looking for. So what do we know about intermediate filaments? They're composed of three domains: a head domain at the end terminus, a rod domain, a coil in the middle, and then a tail domain at the carboxy terminus. So formation of the dimer happens perfectly well with the rod domain alone. Indeed, formation of the tetramer happens with the rod domains alone. But in order to go from the tetramer into an intermediate filament, you rely on these N-terminal and C-terminal domains, and the N and C-terminal domains are of low complexity. They're disordered, they're gibberish-like, they're weird. But with recombinant protein, you can achieve all this. You can put the intermediate filament together perfectly well, all by itself with recombinant protein. So here are my reasons for thinking, why do we want to study intermediate filaments? And so the first point is that the low complexity head domains are critical for filament assembly, and I'm going to show you that. The second is that there's an extensive history of human genetic studies on intermediate filaments. I'm going to tell you about the genetics of one particular intermediate filament, the NFL protein. But most importantly is this last point, and that is intermediate filaments can be assembled from recombinant protein, thus offering the opportunity to resolve the structure of their low complexity domains in the native biological assembly. This was the key difference for me and why we chose to study these proteins. So here I'm just showing some real quick experiments. We've looked at 5 or 6 different intermediate filament proteins and asked what's required for filament assembly. And in every case, the head domain is absolutely required. The tail domain is not. So that clearly tells us we want to study the functional domain, the low complexity domain that's required for function, and that's the head domain. So as we've been worked with these head and tail domains, we've seen that in every single case, the head domain undergoes phase separation, the tail domain does not. So there's a correlate there. Now, here's where we begin to do some functional assays. We can make a hydrogel droplet composed of polymers of the NFL head domain. So it's 100 amino acids. It forms labile cross-beta polymers. We can use Masato's gel binding assay. So we challenge them with GFP length to the full length NFL protein. You can see in the upper left, it's nice binding. If the tail domain is removed, there's still binding. But if we remove the head domain from the protein, there's no binding. So there's some element of specificity in the binding. So the head domain is necessary. But on the right, it's also shown that the head domain is sufficient for binding. So if we simply take GFP and attach it to the head domain only, we see binding. The raw domain only, no binding. Tail domain only, no binding. So there's some interaction in the head domain that is scoreable in this assay. Now what we've done to pursue this a bit more rigorously is we've made hydrogel droplets, shown in the upper left, and mixed them with total brain lysate. So we take a mouse brain, cryomill it, spin out the junk, take the supernatant, bind it to the hydrogel droplet, let it incubate overnight, wash it extensively, and then elute and ask for what proteins in the brain of a mouse can bind to this NFL head domain hydrogel droplet. And the number one protein out of the thousands of proteins in the mouse brain, the number one protein bound to these hydrogel droplets is the NFL protein itself. So the specificity here is extreme. We have extreme evidence of specificity. That's very good news. The bad news is we don't know that the specificity that we're scoring here is of biological significance. So that's where we get to the point of being able to dig into the science much more deeply and carry out kind of what I consider to be the killer experiment, the experiment that could have really put my lab out of business. And it's shown here. And what we're doing here is to segmentally label the head domain of neurofilament, which is shown in red with C13 and N15, and then use N-tene chemistry to attach that isotopically labeled head domain onto the rest of the unlabeled NFL intermediate filament protein. So now we have something where we can specifically visualize the structure of the head domain by solid state NMR spectroscopy. So we carry out this segmental labeling, we compact by centrifugation, and then we work with Rob and Dylan Murray to go about taking solid state NMR spectra. And the next slide shows the result of that experiment. So on the left is a spectrum of the head domain only. So we know what those are. They're cross beta polymers, and they give the spectrum shown in blue on the left. With a segmentally labeled, reassembled biological assembly in the middle, what you're seeing is the spectrum of the head domain in the properly assembled filaments. And what you can see from the overlay on the right is the two spectra are extremely highly related. So these observations make us, allow us to firmly believe that the structure we're studying on the left is present in the assembled biological intermediate filament. And this gives us confidence that what we're studying in the context of structure is biologically meaningful. So the last few slides, I'm going to switch and talk about genetics. So we have function, we have structure, but what about genetics? So there's a wonderful history of studies of the neurofilament protein in the context of a neurological disease called CMT disease, Charcot-Marie Tooth disease. So what's shown in this slide are 6 different mutations that are causative disease. And what's intriguing to me is these mutations all fall into one of only 2 residues within the entire NFL protein. So 3 of them on the bottom map to proline residue 8, 3 of them on the top map to proline residue 22. These are autosomal dominant mutations, which is already intriguing. So they would be inferred to probably be gain of function mutations. But the fact that they idiosyncratically map to only 2 proline residues to me is really amazing. There's something cool going on here. So let's go to the next slide. Oh, I need to go back. I don't know if I can go back. I skipped a slide, but let me just tell you what that slide said. If we make the head domain with those proline mutations, the head domain self-assembles way too quickly. So it's creating cross-beta interactions way better than it should. And that's terrible for filament assembly. So other people had already shown that those mutations inhibit assembly. What we showed on the slide what we found on the slide that I skipped is that those mutations cause the head domain to assemble too tightly, and that's not good. So now on this slide that we have in front of us, I'm looking at proline, and what everyone knows is proline's weird. It's different than the other 19 amino acids. It's an amino acid, and it's the only side chain lacking the amide nitrogen required for Linus Pauline's hydrogen bond networks that allows for cross beta interaction. So proline can't allow cross beta because it doesn't have the appropriate capacity for hydrogen bonding. However, when you mutate proline to any other residue, including leucine, which is shown on the right, you regenerate the ability to do Pauline hydrogen bonding. You have the peptide nitrogen. And so our idea was maybe that's what's wrong. Maybe that's the problem, that proline is trying to stop or limit cross beta interactions. And by replacing it with any other amino acid, you're regenerating Pauline's hydrogen bond network where you don't want it. So the way we've been able to attack this is with an unnatural amino acid shown on the bottom right, which is N-methyleucine. And we've done this with all 6 residues which lead to these mutations, these disease-causing mutations. So what we can do is incorporate the unnatural amino acid. So it has the leucine side chain perfectly well, but the Pauline hydrogen bonding is blocked because that nitrogen atom is methylated. So what we see on the next slide is kind of the technical means by which we approach this, and that is Glenn Lissack and Xiaoming Zhao synthesized a peptide containing the head domain where they could introduce the unnatural amino acid at proline 8 or proline 22, and then put a thioester on the end of that and have a free cysteine on the end, the N-terminus of the remainder of the NFL protein is shown on the top, and then use native chemical ligation to bring the two together. And now you've recreated the NFL protein with a single change, and that is an unnatural amino acid at proline residue 8 or proline residue 22. And in this case, what we've done is to change any of the residues to which the protein is mutated, be it leucine, arginine, serine, to the methylated derivative of that amino acid so that that amino acid could not participate in Pauline hydrogen bonding. So the next slide shows the results of this experiment. That change, that single chemical change, cures or fixes the filament perfectly. So the aberrant filament on the left, for example, proline 8 changed to glutamine, gives terrible filaments. But if we put methylated glutamine at position P8, what you see on the right is beautiful intermediate filaments. So the simple change of that hydrogen bond cures the phenotype. Same happens on the P22 serine mutation on the shown below. And we've done this with all 6 of the mutations. So not only have we looked at this in the context of the intermediate filament itself, but we've looked at it in the context of the isolated head domain. So the head domains, as I told you, when they're mutated, for example, proline 8 to glutamine, they prematurely self-associate, and they're cloudy. They create polymers when they shouldn't. If we replace glutamine at proline residue 8 with the methylated derivative of glutamine, it clears up and the appropriate level of interaction is observed. So let me end by saying that this is not going to be limited to studies of intermediate filament proteins, because the human genetics are really, really helping us, as is the structural biology. So what I'm showing here are 2 recent structures of cross-beta polymers reported by David Eisenberg. On the left is the TDP43 low complexity domain. On the right is the HNRMP low complexity domain. And I'm pointing out the locations of prolines that are mutated, that can be mutated to cause disease. And it's very nice to look and see where are these prolines located. So let me just look at the one on the left and point on the left. The location of that proline is right between 2 short cross-beta strands. So that proline, since it does not allow for Pauline hydrogen bonding, insulates those 2 cross-betas. If, however, it's replaced by any other amino acid that allows for Pauline interactions, then what would normally be a labile interaction composed perhaps of 2 short cross-beta interactions allows for a much more extensive interaction and leads to a pathological association of these low complexity domains that's way more, way stronger than it should be. So let me close with the last slide and kind of summarize our thinking on this. So I'm showing a couple of low complexity domains in the box, and what we're thinking is tyrosine, phenylalanine, and methionine residues favor for non-specific collision of these LC domains. And I think we all agree on that. The phenylalanines and tyrosines and in certain cases methionines can allow for these proteins to bump into one another. What we're saying is the collision facilitates transient adoption of bona fide protein structure, which is kind of shown on the right, shaded in red. We say that the structure is a simple derivative of the chemical genius of Linus Pauling. It's just simple cross-beta interactions, which are used all over the place in the proteome. What we're saying is the cross-beta structures are labile, they're localized, and they're highly specific. And what my prediction is that thousands of these transient structures will be operative in cells all over cell biology. So with that, let me thank my trainees and my collaborators for all the work that they've done over the last decade as we study this. And I admit that we think about things quite differently than other people in the field, but the nice thing about science is that in time, truth will prevail. If we're wrong, we're going to be wrong. If we're right, we're going to be right. So thanks for your attention.
Thank you, Stephen, for very inspiring lecture. And before we end, I just would like to squeeze one question. Can you use the data that you presented as a therapeutic strategy for Charcot-Marie-Tooth patients?
You know, so the answer to the question is I can't think of anything now. So I would like to be able to say, no, this is going to allow us to think about ways of loosening this interaction to correct for the disease. I can't see at this point how I come up with a therapeutic that loosens that structure. But the good news is I think we now understand the molecular basis of the disease. And I think by understanding the molecular basis of the disease, then someone much smarter than me is going to figure out how to go about therapeutically treating that disease. So no, I can't think now how I would do it, but I think what we're offering is an understanding of the science such that someday that might be possible.
Thank you so much. And I would like to thank all the speakers of this fruitful discussion this day. And I'm looking forward to see you all in part 2 tomorrow. Thank you all. Good night, and hopefully fruitful election for our friends in the United States. Bye-bye.
Bye. Thank you. Bye-bye.
Thanks, everyone.